Hello, everyone. Welcome to another episode of the Overcoming Chronic Illness podcast. My name is Dr. Brian Rade, and I'm a naturopathic doctor. And today I am joined by Dr. Penny Kendall-Reed. Dr. Kendall-Reed is a fellow naturopathic doctor, and she developed a number of years ago a genetic decoding software program known as, or report, I should say, known as GeneRx. GeneRx is a report that looks at a number of different genetic SNPs, single nucleotide polymorphisms. We'll get into the jargon if you're not familiar with genetic reports or what SNPs are. I'm sure we'll get into that in the interview coming up. Um, and I was really to be perfectly honest, when I first heard about, first saw one of her reports, I didn't know that she had. I saw the report a patient brought it in. Didn't know exactly who, didn't realize that it was, one of her reports. I was like, it's another genetic report. There are so many companies out there that are offering these reports. Once upon a time, there were just a couple of options. Now there are dozens of different options. And quite frankly, I've seen a lot of these reports from a lot of different companies, and a lot of them are, in my opinion and in my experience, not really all that useful. They'll have these, reports that look really nice. The graphic designers did a great job. Then you look at what they're reporting on, and it's either not that many genes, or the genes that they do report on, they make these grandiose claims saying like, oh, you've got this polymorphism here, this polymorphism there. And that means that you should be doing this with your diet. That means this about your microbiome. That means about this or that supplement or whatnot. And when you actually dig into the research literature, there's very, very, very little research. It might be just on, some, animal studies, or it might be, they're basically making a lot of extrapolations. So when I first saw one of the GeneRx reports, I was like, oh, all right, it's probably another one of those reports. And so, took a look at it. It's like, oh, it's actually reporting on a lot of genes that are not commonly talked about. And so, one of my colleagues actually did a really deep dive into her reports. And, well, I started poking around at a few genes and saw that, like, oh, there's actually a lot of research, like a lot of human clinical trials, sometimes like gigantic, like large scale human clinical trials looking at the relevance of these genes. And so I asked one of my colleagues to, look into this more. She, went through the entire report, and there's just oodles and oodles of research backing up what is in this report. So I was really encouraged, started looking into it in more detail myself, took a look at my colleagues' write up on that, and it was, I was very, very impressed. So I started recommending this for my patients, and I reached out to Dr. Penny Kendall-Reed and asked if she would be willing to chat with me on my podcast. She graciously said yes, she would, and so I'm really excited about having that chat. So we'll get into the obviously all the weeds of it coming up soon, but I think that there's a lot in those reports that are a lot in her reports that are certainly relevant to folks with complex chronic illness. Things related to inflammation levels, blood sugar regulation, metabolism issues, etc. But then also, I think there's a lot of really useful information in there for just folks in general. So if you're listening to this because some of my podcast episodes are maybe a little more biohacking focused, or if you were once suffering from complex chronic illness, you're a lot better, all the way better, and you're just wanting to health optimize, there should be, I think, something in here for just about everyone. And I'm really excited to get into it soon. So I will pause the recording, and I'll be joined again in just a moment by Dr. Penny Kendall-Reed. All right, everyone. Well, welcome back to the podcast. I'm now joined by Dr. Penny Kendall-Reed. Dr. Kendall-Reed, thanks so much for joining me today.
Thank you for having me. It's a topic that's near and dear to my heart, so I'm excited to share everything with it.
Awesome. Well, looking forward to it. Well, just before we jump into the, nitty-gritty of, what are SNPs and genomics, and, some of the ins and outs of the report that you offer, would you mind just telling folks who you are, and just like a little background on who you are and why
Yeah,
how you got into this?
absolutely. So, I'm Dr. Penny Kendall-Reed. I'm a naturopathic doctor in Toronto, Canada. Gosh, I've been in practice for way too many years, almost 30 years, and love it. Feel so blessed to be in naturopathic medicine. It and genetics takes it to an even bigger level, because as our core as naturopathic doctors is get to the root cause of the problem, right? Don't just look at the symptoms, get to the cause of it. How much closer can we get than our genetics? We see our potential. Then you marry it with symptomatology and see which gene is turned on or turned off, and we'll get into all of that. But it literally is genetics to me is like an intracellular MRI. It's like I get to see what's going on that little level. And I first got into it probably 19 or 20 years ago now, because I would see two patients come through the office, same symptomatology, similar backgrounds, and you probably see this all the time. And the same protocol would be fantastic for one person and it would aggravate another person or just not do anything. And I would say, well, like, why don't you fit in the box? You have all the symptoms. Why can't I just tick that off? And more of that started happening. And then I just read an article on genetics one day, and that started me down that path. And I spent many years researching it before I even tested my own genes because I wanted to know, well, is it valid? Is there a point in this? Are you just going to tell me something scary and then go, good luck with that? There's nothing you can do about it because I'm not interested in that. And I'm not interested in that for a patient. But years and years of research went down the road, and then I started testing different supplements and nutrients and things. And did that make a difference with that gene? And here we are many years later.
Cool. Very cool. Yeah. So you were basically into genomics before it was cool.
Yeah, exactly.
Awesome.
Yeah.
That's really great. Well, just I know a lot of the folks who listen to or watch these episodes of the Overcoming Chronic Illness podcast, they're pretty well educated. They're, it's not their first rodeo. But that being said, genomics is like it's a little bit of a special category or topic, I guess, in that it's a little more maybe heady or a little more nuanced or detailed than maybe certain other topics. So just to make sure we're all on the same page, would you mind just giving us a quick little crash course and like, what are genomic reports actually looking at? Like what is a SNP? What's the relevance of looking at these things? If you could give us a little crash course in that, please.
Absolutely. So the best way sort of describing it, if we think about our gene, we know that we take half a gene from mom and half a gene from dad, and that makes up our gene. And we all sort of understand that concept. That half a gene is called an allele. So we take one allele from mom, one allele from dad, and that makes our gene or per se, and tells us how we're going to code for it. However, every time a cell turns over, we should have those same two alleles being turned over. It should make an identical replication of itself. Sometimes it replicates into a different gene, or sometimes it can have a big stressor on it, so it replicates into a different shape or a different coding. The purpose of a gene is to make a protein. That protein can do something good like increase your metabolism or make serotonin or happy hormone. It can do something bad like increase inflammation or push estrogen to a cancerous form. So that those two alleles, they determine the recipe, if you will, of that protein that's going to be made. Every gene has a very specific recipe. A SNP, though, is when that recipe gets changed. So that basic functioning of the gene has a different recipe, and now it produces either too much inflammation or not enough serotonin. So the purpose of testing genetics is what's inside your cells, what possibility of inflammation or serotonin, etc., and all the different pathways do you have? And then what can we do about it? If it's too much, how do we bring it in? If it's not enough, how do we boost it up?
Perfect. Great. Thank you. It's like you've said that before.
Yeah, once or twice.
Once or twice. Um, and then could you just, before we kind of get into, some of the more specifics of what the report looks at, could you give us just a little backstory in terms of how this test was developed? Like I know you've been in this for
Mm,
quite some time, but like, how did you go from like, kind of dabbling 20 some odd years ago to, like now you have this GeneRx.
absolutely. So after I finished testing them on myself, and literally genetics saved my life. So it's something that I do hold dearly, and tested it out on family and friends. That's when I started using it with patients. But at that time, there wasn't a platform that I could upload a testing profile like a 23andMe into and get the information that I needed. And the information I need is the interaction between genes and what each gene meant once it was decoded. So I used to spend 7, 8 hours per patient pulling out gene by gene from 23andMe, which one I wanted. Then I would write a blurb to that patient. What does this gene mean and how it interacts with the next gene? And what are we going to do about that? And marry it to their symptoms. And about a year and a half later, I was just way overworked from spending too many hours on this. So I created GeneRx, which is the company, and it's the first algorithmic program. And that part was really important for me because all of the testing platforms that are out there, they treat gene by gene. So a really good example there is, let's look at carbohydrates. There are three really important genes which talk about how much insulin you make to carbohydrates, but also do you create inflammation in your GI tract through carbohydrates? Do you feed cancerous pathways through carbohydrates? Like they each have different points as well. Treating gene by gene would say, let's say you code perfectly for the first gene. Great, you get to eat carbs. It'll say you can have carbs. And then the second one, let's say you're heterozygote for that gene. So you have one normal allele making that normal amount of protein, one variant allele making less of that protein. Oh, you're not so good with carbohydrates. You need to eat less carbohydrates. And then let's say the third gene, you're variant for. Then it's going to say you can't have carbohydrates. Well, what does that mean? Do I get them? Do I not? You have to integrate them all together. And what does that mean? Are you creating inflammation? Do you stimulate cancerous pathways? You can have this exact amount of carbohydrate per meal. And that's the sort of protocol that I wanted to create across all of the genes for people.
Um, so you've kind of touched on this next question already, but I'll just ask you more straight out here. How does the GeneRx report differ from other reports, like over and above the algorithmic kind of component? And what other ways does it differ from other reports?
For sure. So it's long, it's intense, it's like 65 pages.
Mhm.
It gives a really good definition for us of each gene. So if somebody's new to genes, if you can really learn about that gene because you're not just getting a one-liner about it, and then it'll tell you key points with the variant gene. But the biggest part is it gives you full treatment protocols. So after it does the integration of different genes, it does a full explanation of what that integration means. And then what do you do about it? It gives you an 8-week protocol because it takes about 8 weeks to reset a gene. So it could be this supplement at this dose for 8 weeks, and then we get to drop the dosing down to this for another 8 weeks, or you can come off of it, or you need to stay on it at that lower dose. Um, the report is also editable. So in each treatment section, let's say you wanted to put somebody on something to boost up serotonin, but they were already on a pharmaceutical. So this is where you can edit the report as a healthcare practitioner and say, I know that we don't need to do this. You're already on it, or this is why you're actually on this medication, and it is well suited for you. You can change dosings for patients. You can change companies if you want to use a different company in terms of the supplements. It's a very user-friendly report. So it gives you the full treatment programs, but then you can also just add in little details for your patients.
Great. And since you haven't said it because you're probably just being kind and diplomatic and Canadian like I tend to be, I will just say that having looked at many other reports out there, what I was really impressed by, I mentioned this in the introduction, but just going to say it again because it just really warmed my heart when I learned this, that your reports are just very like heavily evidence-based, like just ridiculous amounts of research backing up each of these SNPs, single nucleotide polymorphisms that are looked at. So that is something I would say that really notably sets your report apart from a lot of other ones too, if I may say.
Thank you. Yeah, it's something that's very important to me. When I was doing the research for each of the genes, people often ask me, how did I choose the genes and how did I know which were clinically relevant? I had a rule where I would need 7 different studies or 7 different sites with clinical research behind it that all said the same variable about the gene. Because we all know that we can get one study and we can make a study say pretty much anything that we want. So I needed 7 independent studies that were all saying the same thing, not just that Snipedia had a slew of them. It had to be very sort of sound research papers and all with the same messaging system. And then that's when I would take into credence. Yes, you can't just go by the number of people that have that SNP. It has to be clinically relevant and it has to be the correct RS number for that gene. So I spend a great deal of times that I'll often get docs asking for the actual studies. And I have a document which is over 68 pages of just lines, and the lines are just links to research paper. I mean, there's so many research papers that went into it, and then I continue to update it. As new information comes out, I'll change the algorithms. I will change. I shouldn't say that. I've added new algorithms. I haven't changed any of the older ones, but I've changed treatment protocols. As new products come out, as new research comes out, I'm always updating the program. And if I add in new genes, the reports automatically propagate those new genes in the treatment protocols as you go along. And so it's a very user-friendly and scientifically grounded platform.
It's really great. And yeah, very, very refreshing in this field, if I do say so. Um, so as far as the application of this report goes, I mean, I kind of mentioned also in the intro that there's, in my opinion, to my understanding, and you know better than me, which is what I'm going to ask you. Um, so I think that there, to my understanding, there are applications to folks who are dealing with complex chronic illness, whether it's fibromyalgia, chronic fatigue syndrome, autoimmune conditions, maybe mold illness, chronic infections like Lyme and whatnot. But then there's also, I think, applications for just all of us in general, because like we all have a metabolism, we all have, cholesterol levels and blood sugar and all these things. And so I'd like to ask you about sort of the application to certain conditions, but I'll just kind of like phrase the question or pose the question, but then that's kind of the ultimately like folks listening to sort of have a sense of, okay, if I have X, Y, or Z condition, how might this apply to me? But without, having 3 days to talk to you nonstop and going through like every, known medical condition under the sun, maybe we can kind of just generally categorize them sort of. So I guess for folks who are dealing with, say, I'll just start broad. Folks who are dealing with complex chronic health issues, so fatigue and or pain and or neurological symptoms, a lot of inflammation, maybe related to things like mold, Lyme, other co-infections, viruses, SIBO, etc. Like just to kind of imagine like your I say this affectionately, but like your classic like dumpster fire patient who's got everything under the sun and every symptom under the sun. What elements of the report would you say would be the most useful or relevant to a case like that?
Absolutely. So initially, I think what would have got that person into a problem would be the inflammatory genes, the detoxification genes, and the mitogenic genes, along with autophagy. So um, genes, as mentioned, they interact with one another. So the inflammatory genes, right off the bat, if somebody is variant for those genes, they're making up to 85% more inflammation every time it is called upon. So let's say they are exposed to a mold or they're exposed to Lyme. Their inflammatory load, rather than being this, is this. And then that inflammation begets more inflammation. Inflammation, like cortisol, turns 90% of genes to their adverse position. So let's say they had a weaker stress gene here. Now, that inflammatory load is now going to turn on the stress genes. And then the stress genes augment the inflammation again. Detoxification, if they have poor, particularly, oh no, if it's too fast for phase one and too slow for phase two, that's the most inflammatory state somebody can be in. So if someone's exposed to mold or toxins or a bacteria, and that toxin goes intracellular or pathogen goes intracellularly, too fast phase one means they're pulling it out really quickly and dumping it into the extracellular, where phase two should then pull it out. But if phase two is really slow, it can't get it out. So now you've just dumped it from being quiet inside the cell to outside the cell where it's active, and now it can't get out. And that's where we see these massive differences between two people being exposed to a mold or a toxin. They have a hyper-inflammatory reaction to it, and then they can't get it out of the body. And it keeps creating caveat after caveat after caveat. That then starts to trigger their stress genes, which also impairs their sleep genes. So now they don't go into deep sleep, and they have decreased energy levels from both of those pathways. Those are all the offshoots, but for most chronic issues, it begins in those inflammatory, mixed up phase one, phase two detoxification states, and then the mess occurs. It can occur from low autophagy as well. And it can occur, particularly what I saw during COVID. There's a gene, IR5, which talks about how quickly does your immune system see a virus or a bacteria, jump on top of it and then start to destroy it. Some people, if they're variant for that gene, they don't see it quickly at all. So that pathogen comes into the body, it's invading, it's now replicating, and your immune system is just kind of hanging out. And then suddenly the immune system goes, oh God, there's something in here. I've got to do something about this. But by that time, the pathogen has taken hold. And those are the people, again, comboed with high IL-6, because we know COVID really targeted IL-6, had such bigger effects. And this is why I love genetics, because you can see the potentials in all people. Almost all disease comes from an aberrant pathway that is in GeneRx. It's not going to jump out and say Lyme, but you're going to look at the pathogen associated one. You're going to look at the detoxification one. You're going to look at the inflammatory one. You treat those guys, you're treating almost all of the side effects with it. Again, mitogenesis or mitochondrial function can have an effect there too, but it's usually secondary to the mold or the pathogen itself.
Um, could you speak a little bit to the kind of mitogenesis related genes?
Yeah, absolutely. One of the biggest ones is SOD2 or superoxide dismutase 2. So this is a phase 2 detoxification gene as well, but it's right within the mitochondria itself. So it's decreasing the production of inflammatory markers or ROSs and cleaning out the mitochondria so that they can keep replicating themselves as well as keep making ATP to have every function going. There's also SIRT6 and BDNF, both which also play a role in mitochondrial function. They're a little bit more specific for neural repair. So when they're low, that's really involved in a lot of the different neurodegenerative diseases, Alzheimer's, dementia, Parkinson's, MS, etc.
So with SOD2, would that be primarily related to keeping up that kind of balancing out like all the pro-oxidants that are, or the pro-oxidative pathways that are involved in ATP production, like trying to keep those neutralized so that, I always explain it to my patients as mitochondria being little combustion engines that are like doing
Mhm.
work, like making energy, but you would never run like say a gas powered generator in the middle of your house because you would kill yourself from all the exhaust. So we need to neutralize that exhaust, preventing, lipid oxidation and or peroxidation and things like that. So with this, the SOD2, is that primarily, is there some other function that it serves over and above neutralizing free radicals in the mitochondria?
Yes, it actually directly increases mitogenesis. So the replication and the repair of the mitochondria themselves outside of ROS reduction and oxidation.
Okay, great, cool.
Yeah.
And then just for folks listening, so talking about like, okay, so someone's listening thinking like, okay, like I'm that, aforementioned dumpster fire, and I've got all these things going on. And okay, so I do this test and I find out that I've got imbalances with my phase 1 and phase 2 detox and my mitogenesis abilities and all these different things. So are there things that I can actually do about it? Or and
Yeah.
not that we need to get into specific protocols. And sorry, I meant to mention this earlier. As per usual, we're not offering any medical advice during this podcast. This is all for informational purposes only. If you need medical advice, please talk to your healthcare provider to get that advice. So not getting into specific protocols, but could you maybe give us like a hypothetical example of like, okay, someone's got all these symptoms. They've got maybe a phase 1, phase 2 imbalance, or they've got a mitogenesis issue, around SOD2 or SIRT1 or whatever it is that happens to be. Could you give us an example of like what they would actually actionably do with this information?
Absolutely. So let's say their phase one is too fast, as mine is. Curcumin will actually slow it down. So curcumin slows down phase one detoxification. Really important because if you're already slow for phase one detoxification, this is just a little side note, curcumin is bad for you. It slows down your slow down phase one too much, and that's actually pro-inflammatory. For myself, or someone whose phase one is too fast, it's a fantastic anti-inflammatory and it slows down phase one. So does caffeine. Coffee, yay for me, good for me. In fact, 2 to 3 cups of coffee a day will reduce my risk of having a heart attack by 68% through regulating phase one detoxification. We can upgrade mitochondrial output, SOD2 specifically with astaxanthin or NAC, glutathione, lots of different ways that we can, upgrade that one specifically. We can even a ubiquinol. What I'll often do is try to cross cover multiple genes with less supplements. So NAC and ubiquinol, or NAC and glutathione, they will upgrade SOD activity, but they'll also upgrade GSTP1 and NQO1, the other phase 2 ones. So that way we can cover more with less things. To upgrade mitogenesis, we can also use resveratrol, and then we can also use spermidine. We also, for these patients, though, we have to look at, because by the time they come to us, and I know you see this too, it's not just their inflammation and detoxification that's off. Those pathways have now triggered their adrenals. So we have to look to their stress genes, and trying to treat these patients and not treat their adrenals or their stress genes at the same time, or all the offshoots, they will not heal. So yes, we see the cause of it in the report, but then you also see all the potentials of where it can go to. Particularly, so we'll look at the sleep genes. How well do you transition into deep sleep? Do we need to fix that as well? Because that can also be a big part of it. And we can easily reset stress genes with a casein decapeptide and a theanine combo. We can easily reset the sleep, even just by using high-dose melatonin, comboing it with some passiflora, depending on which those genes are. So we have to make sure that our healing pathways are also reset, not just our pathological pathways that are off.
Mhm. I'm just curious with curcumin, is that so that slows down phase one?
Mm-hmm.
Is
Which is
that
great for me, but
right?
not great if you're slow.
Right. So, is that largely because it just like preoccupies your cytochrome P450s? Is
100%.
a lot of
So
work for them?
yeah, it's decreases the amount of cytochrome P451 1A2, the main enzyme that needs to go inside of it, bind to that toxin and take it out. So you're leaving intracellular toxicity here, and if you're slowing that down even more, the inflammatory load gets higher and higher and higher.
Mhm. And sorry not to belabor the point, but is it just because the curcumin just um, preoccupies a lot of the CYP450 resources, or is it that it actually
It
downregulates the
actually down regulates the CYP1A2 specifically.
Okay,
Yeah.
cool. Okay, interesting. Does it use, I think it uses 3A4 as well for its metabolism, but
3A4 as well, 1A2 massively, 3A4 it does. 3A4 is not quite as strongly associated with curcumin as the 1A2 is, but 1A2 for sure.
cool. Um, I'd like to ask you about a few other applications or conditions that might benefit from having a GeneRx report done. But anything else you'd want to add as far as the like more like complex chronic illness applications would go? So I think it's been quite a bit, but you know it better than me. So
Yeah, no, again, like when I'm doing anything with a patient, I'm sort of also going back to our naturopathic roots of diet. So diet and exercise, because we all can understand how there's not one diet for, keto can work for weight loss for somebody, and high carb, low fat can work for somebody. And now we know why it's genetics and this will dictate it. But diet can be just as damaging. So, we always want to go back to that. And also exercise, doing the wrong form of exercise can also generate a lot of toxic byproducts and inflammation, etc. So knowing those two basics, just sort of getting back to our naturopathic foundation there. Also, I'll always include those no matter what the case is.
okay. And just kind of a related question, just a little bit of a side question out of curiosity. So I know in my practice, I've had, many patients that have come to see me over the years and, just where, I have a reputation for treating folks with, chronic infections and mold and heavy metals and all the things been mentioned. And, in many cases they come in and we test them and they have those issues and we treat them and they feel better. I've also had, quite a few patients though over the years. I haven't been at it for 30 years. It's only been 16 for me. So I'm halfway there. I'll get there eventually. But I've had a number of patients over the years where they come in saying like I just had a new patient actually very just yesterday, I think, or the day before yesterday, and she has seen like all these great doctors, these like really, very well versed, very smart functional medicine docs. She's been being treated for like 8 years or something like that for Lyme, molds, everything under the sun. She's had other genomic testing done, whatnot, every supplement you can think of, fancy pants, like more newfangled things, like, well, kind of old. I was going to say methylene blue is not newfangled.
Methylene blue, yeah.
Drug that was, I think, approved by the FDA, if I'm not mistaken, once upon a time. But
Mm.
anyways, lots and lots of different things, and yet, still having symptoms. And in a case like hers, where we're going to do some additional testing, see what's going on. But in cases where it's like patients are saying, well, I've got all these, infections and this and that, but like I've been treated for years and I'm still not better. Maybe I've hopefully improved somewhat at least, but like I'm still not better. And so like, I've had Lyme for 10 years. I've been treated for, ever many years. So if a patient like that shows up at your doorstep,
Mhm.
I'm wondering how many times, or have you seen cases where, they've had all these diagnoses, all these things they've been treated for, they're still not better, and then you find something within the GeneRx report, or some multiple things within the GeneRx report, where it's like, okay, the things that actually finally cracked that case were that it was about figuring out the phase one, phase two imbalance, or it was, getting them on the right type of diet that was actually designed for their individual, genetics, or, something else that you found within that report that had nothing to do with that magic antibiotic or that magic binder for mold or that magic nasal spray or whatever it was. So hopefully I articulated that well enough.
Yes, no, perfectly. Actually, do you see, I do a lot of work with a medical doctor in the US, helping her, like I'll go through her patient's genetics for her in exactly the same arena that you're in. And generally what I see with those patients is it's really slow phase one.
Hmm.
And they are on curcumin. They're slowing it down further because everyone thinks it's this great anti-inflammatory for everything across the board. So they're not actually doing anything beneficial, or they may be increasing phase one, but if they have the curcumin there, it's a moot point. They're also hypermethylating. And I see this, and this is a big pet peeve of mine, is people methylation needed. It is a catalyst. It speeds up reactions. And it's one of the first areas within genetics that was sort of brought to the forefront. So everybody loves to focus just on methylation. Um, you can't specifically turn on methylation though and say, you know what, I would just like to upgrade that pathway and not that one and that one. You turn on methylation, it's like the Christmas tree. Every light bulb goes on. So if you do that before you turn off the stress pathways and the inflammatory pathways, all the studies show us that you upgrade those pathways even more. So you're making those pathways way worse. And every patient of that description I've seen is on methylators. So we have to pull them off, and I pull every patient initially off of any methylator, particularly if their methylation genes are strong. I had a patient today, there's a cancer patient who is put on so many methylators, and she got perfect methylation. So she's just hypermethylating from day one. So I see that as a big issue. I also see the stress genes, no matter what their position, even if they're slightly weak or really weak, that is a big blocker for those patients because, as mentioned, stress and inflammation block the treatment of over 90% of genes. So you could do everything perfectly in every other section. And if you don't properly treat stress and inflammation, or you leave one area of them untreated, it's going to stop all treatment. So we need to not just decrease the excess release of cortisol, adrenaline, and noradrenaline with our adaptogenic herbs. We need to stop the blockage of the stimulation of the stress pathways at the hypothalamic and pituitary level. And that's with like the casein decapeptides, et cetera, that unblock those receptors, turn FKBP5 off, the gene that makes the protein that's blocking those receptors. And then we also have to look at all forms of inflammation. So diet, as you spoke about, GIPR. Are you making a ton of inflammation in your GI tract through disaccharides or carbohydrates? So if you're not addressing that side, here's another version of inflammation. If you're not going into stage 4 sleep, even sleeping 7 versus 8 hours a night, starts to increase inflammation in 5 days. So just that alone. So we have to treat all of the areas of inflammation and all of the areas of stress. Watch out for phase one detoxification with that chronic patient and watch out for hypermethylation.
Are there genes in your report that um, indicate that there is hypermethylation?
No, just tells you how you methylate, but the report will say, if you're perfect, don't do this.
Hmm. Makes sense. And I know you've mentioned the casein decapeptide a few times now, bearing in mind that this is not a CME accredited podcast episode, so I'm assuming we're talking about lactium.
Yes, we're talking about lactium. So either serotonin plus or stress reset.
Okay, great.
Yeah.
And
Sorry, I keep forgetting. I
no,
switch
no,
back and forth
it's
between CE and yeah.
yeah, yeah, no, I appreciate it. I do the same thing, but
Yeah.
um, so with lactium slash, I think casein decapeptide sounds cooler, but anyways,
Yeah.
would you mind just because I think a lot of folks are not aware of the existence of that, at least here in Canada, anyways. I don't know. I'd never heard of it until I had a patient who had consulted with you, and I was like, oh, what's this stuff? And I was like, again, I don't know, maybe I'm getting skeptical in my whopping 16 years of practice, but. I was like, I wonder if it's just, it's like, wow, there's so much research on this thing. Like it's
So much
so
research on
cool.
it.
Yeah, it's so cool. I'm using it all the time now. But
Yeah, it's phenomenal. So it's a casein decapeptide. So it is a 10 amino acid sequence, which is isolated from milk. It's lactose free. For anybody who is allergenic to milk, it's still fine because the immune system can only see something that is 10 amino acids in length. Or sorry, 12 amino acids or longer. This is 10 amino acids. So it kind of scoots under the radar of the immune system. So even my anaphylactic dairy patients have no issue with it. What it does is, so for listeners, and I know that you know this, but just for listeners, just in case, when we have a stressor, our adrenal glands make adrenaline, noradrenaline, and cortisol. Once we've dealt with that stressor, those hormones bind into receptors in the hypothalamus and the pituitary, and that's a signal to the nervous system to say, great, I'm done with this stressor. Tell my adrenals to stop making more stress hormone and take me back to campfire parasympathetic. We need that binding of those neurotransmitters into those receptors in order to stop making more stress hormone. If that messaging doesn't happen, your adrenals don't know that they can stop making the hormone. They just keep making more and more and more. So there's a gene FKBP5 which makes a protein which blocks those receptors. And then there's another gene which tells you how many of those receptors you make. But what lactium does is it goes in and it kind of kicks out FKBP5 out of those receptors. So now they're open up again. So now we can have our own hormones come in and turn off, engage negative feedback in that HPA axis again. At the same time, it turns off the FKBP5 gene. It binds into the GABA A1 receptor immediately, which is the calming receptor in the brain, but it doesn't bind it to the GABA PBR receptor, which is the sedating receptor. So a benzo binds into both of those. This only binds into the calming one. And there are zero side effects. We isolated it, we've discovered it in breast milk, that babies that suckled with mom, their cortisol levels dropped. And they thought, well, is it because they're suckling with mom or is there something there? So they put formula in a bottle, breast milk in a bottle, and regular milk in a bottle. The breast milk baby, their levels dropped most rapidly, started playing around with it in the lab, and lo and behold, there's the sequence.
um, so with the lactium, is it how long does it typically take to see a response in a patient who's taking that?
Honestly, 48 hours. If you get the right dose, what you have to do is get the right dose. Most people do one a day. No, no, no. I'm generally starting everybody at two twice a day, usually, unless, and then how quickly they drop down is according to their genetics. Or they take it with food, and then it's going to be completely blocked. You have to take it empty stomach. Um, that's like if you take it with food, it's like saying, I'm going to increase eggs in my diet, so I'm going to eat cake. Yep, there is an egg in cake, but we don't eat cake and our body doesn't go, "Ooh, egg to go do this." You have to take this protein peptide on its own away from food. We can mix it with other empty stomach supplements, but if you mix it with food, it just gets lost in the food as like a little bit more milk. So it has to be empty stomach. But generally, I see most people start to feel a difference in 2 days. unless there's a great deal of inflammation in the body blocking receptors, and then which case 10 days to 2 weeks. I've never not seen somebody. But don't forget that inflammation makes a big difference with anything binding, particularly in the brain. And that's because of that lock and key fit, right? So every neurotransmitter has a lock and key fit with its receptor. If this is the receptor and it's this shape, inflammation comes along and makes it look like this. Doesn't matter how much ingredient you have to go in here, it can't get in there anymore. So you got to open up the receptor by reducing the inflammation, and then stuff can go in.
Um, with Lactium on an empty stomach, I'm just thinking for folks who are taking it at nighttime to go to sleep, like, is it okay to, like, would taking melatonin or theanine or other things like that, would that interfere or that's
No, absolutely. In fact, there's some great products that put all of those together. So yes, empty stomach is just 30 minutes or more before food or 2 hours or more after, but it can be with other empty stomach nutrients, no problem.
okay. I think we should probably move on from the complex chronic illness patient, just because I want to ask you about a couple of other things here, unless there's any final thoughts that you wanted to share on
No.
that. Okay. Um, so yeah, one of the things that I was really fascinated with the GeneRx report when I dug into it was just so many SNPs, like so much research literature looking at some of these genes as they relate to challenges with weight loss. And
Mhm.
one of the reasons that I have premature gray hair is that I've just had so many patients over the years where like they've done everything right and they cannot seem to lose weight. And like patients who are like, fasting like crazy, like, carnivore diet, keto diet, they'll swing the other way, like just going full vegan, like exercising regular, like just still can't seem to lose weight. So thankfully that's not a majority of patients, but like they're out there as you're
Absolutely,
nodding
they are.
knowingly. So could you speak a bit to some of the things that you've seen in practice in terms of using the data from the GeneRx report and how that's translated into cracking some of these like very challenging like weight loss resistance cases?
100%. There are so many different weight loss books out there. Like you go into a store, there's just like rows and rows and rows and rows. You go onto Amazon to try to look up one, and there's bajillions. They all work for somebody. They don't work for all of us. I've written many of those books. I don't even keep them around anymore because they're obsolete. It all comes down to our genetics. So our metabolism is almost entirely controlled by three hormones: leptin, adiponectin, and ghrelin. They ultimately dictate how much insulin you're going to make. They dictate GLP-1. They dictate all of those guys. Our hunger, our satiety, our rate of lipolysis and lipogenesis. So how much fat we're breaking down and how much fat we're creating. Those three adipokines or those three hormones, they control the majority of it. GeneRx has 6 different genes which talk about how much of each of those you make and in different scenarios when you're on a weight loss diet, when you're not, when, etc. So just in terms of getting the metabolism working, you have to know how much of those hormones you're making and then push it one way or the other. A perfect example, I am what's called FTO variant. So I have the slowest metabolic rate through one of the biggest metabolic genes called FTO. I have always known that I had to eat less than the average individual. I required less calories. I felt absolutely fine. Didn't mean it was fun, but I always needed less to maintain a specific amount of weight. Now, I was very blessed. I grew up in an incredibly healthy environment. My mom fed us organic, that's 50 years ago, more than, etc. So I was exposed to a very clean diet that didn't fully trigger my FTO gene to its on position. Now that I know it, I've retrained that gene, kept all of my metabolic genes in their off position, so that I don't do this. If I eat what my husband eats because he needs carbohydrates, my genes don't, I will start doing this. Um, I need more fats than he does, not ketogenic. So everybody's different. But your genes will dictate how much insulin you are making um, to carbohydrates. It will dictate from saturated fats. At what gram level do you start kicking in fat storing or increase inflammation? What are your hunger cues? What are your satiety cues? How quickly do you break it down? And you can't guess it. Especially when somebody goes on a ketogenic diet at first, they cut out all sugar and then they lose weight. And then they're like, oh, this is it. This is the one for me. And then, like 3, 4 weeks into it, it starts to slow. And then they hit that plateau at like a month, and then it can even start to go up. And everybody will lose weight and feel good from pulling out sugar. The question is, are you supposed to do that for long term, and at what extreme? Doing an extreme in any diet increases the stress hormones, and the stress hormones themselves will make you put on weight. So you have to know where where your diet is at in terms of fat burning potential, satiety, and hunger cues. What's going to increase or decrease inflammation? What is going to increase or decrease cortisol? Not everybody's designed for intermittent fasting. If their metabolic genes are really strong, fasting is actually a stressor on their body and increases cortisol. So the report gives you all these clues into your very specific genetic diet. Some people are way overloading protein. That's just turning into inflammation. So it's very individualized.
Would you be able to give us maybe an example from your practice or some example of what that might look like for someone? Like just kind of maybe an example of someone who was you were working with, they were having a heck of a time losing weight, and then like what an actual protocol looked like for them, like give or take.
Yeah, absolutely. So I had one individual who was put on a GLP-1 agonist. So she was about 200 pounds overweight. She wasn't responding with the GLP-1 agonist. Didn't change she was on a ketogenic diet while she was on this. We did her genetics, and her metabolic genes were actually quite strong. Um, she was catered towards a higher carb, lower fat diet. The problem is that she had had an awful lot of trauma as a child, and her stress genes were really weak. And stress will take a good gene and turn it to its off position. So her strong metabolic genes were in the off position.
Hmm.
So I actually didn't need to play around with her adipokines, her ghrelin, leptin, and adiponectin. I needed to change her diet to a higher carb, lower fat. Needed to actually pull her off the GLP-1 agonist and fix the stress genes. And she's actually lost I just saw her last week, 230 pounds. So even more than she
Oh,
had wanted to.
wow.
But other individuals who have come in and they've tried every diet under the sun, but their FTO and their MC4R genes are turned on. And particularly with MC4R, FTO, you can diet your way out of FTO and turn that gene off. MC4R, you cannot. Once that gene is triggered, you cannot turn it off without a supplement. So for that individual, I actually didn't change their diet at all. They were eating perfectly just by chance to what their genetics wanted them to. All we had to do was put in something called TMC, or trimetabolic control, which is a L-carnosine, not carnitine, berberine resveratrol combo, which resets adiponectin, leptin, and ghrelin, but the MC4R gene. And all of her weight started coming off for the first time in years. So it can come from a lot of different places. Um, but don't underestimate the power of stress to turn off a good gene just as much as it can turn on a bad gene.
Very, very interesting. Okay. Um, anything else you want to share on the weight loss front before we move on to another?
Yeah, also, again, look to the inflammatory and detox genes. Inflammation and toxins are stored inside of our fat cells. And when that level starts to rise up, the fat cell, no matter what you do, even if you're treating all the other sections, won't give up any of the fat around it, because the fat or adipose itself acts as a buffer to the toxicity of the inflammatory substrate or the toxin inside of it. So it actually wants to sort of hold on to it. If you do a crash diet, and then you release that fat cell will die. So then it quickly usurps even more fat, and it rebounds back up. So opening those pathways is also really important.
Okay. Um, is it easy? Knocked over my mic. Sorry, one sec.
Yeah.
Okay. So Is there an application of the GeneRx report to folks who are dealing with maybe I'll say kind of like idiopathic dyslipidemia, and translating that for people into English. So there's some folks where they seem to be, eating a reasonable diet. They're, exercising regularly. They're not, holding on to like significant amounts of excess body weight, etc. And yet they seem to have, well, they have elevated cholesterol levels, elevated LDL, total cholesterol, etc. Sometimes, and maybe, so I'm wondering if the GeneRx report has an application to folks like that. And
Mm.
then maybe a follow-up or a second a related question to that would be some folks who have elevated cholesterol, like total cholesterol, maybe LDL's up a bit, but then their HDL is also really quite high. Like say their HDL is maybe like a, you know, 2.3 or 2.5 or something like that, where if only their HDL was lower, their total cholesterol would be in range, somewhat ironically. It's like there's too much of the good stuff. So have you seen cases like that as well? And does the GeneRx report have an application of those cases?
Yeah, so there is actually a whole cholesterol section within the GeneRx report, and the genes there talk about how many receptors you have on the liver to break down and recycle LDL. So the true idiopathic cholesterol is not so much about the formation of cholesterol, it's about the inability to break down the cholesterol and recycle it. And that's with less of those receptors. And then there's also one that talks about how much of an enzyme do you make in order to break it down. So you need both a receptor and the enzyme there. For those people, we need to most often use a red yeast rice extract, which will function similarly to a statin in terms of HMA-CoA reductase, but without any of the sort of inflammatory or myopathy side effects. Most often, I have over the years seen a few people with the myopathy side effects with that. Cortisol also has receptors right on the liver too to increase LDL production, which is why you'll often see even in people with great diets, LDL go up right after a stressful period of time or a prolonged stressful period of time. So again, don't forget about those genes, not from turning off the cholesterol genes, but just from a direct receptor point of view for those people. Then again, we look at the saturated fats. So that's where the dietary part comes in. And some people, particularly for ApoA2, if they're variant for that gene, this is the gene which says when I'm eating saturated fats, at what threshold do I then start making cholesterol and inflammation and store fat and etc. That's at 22 grams. So 22 grams can be pretty easy to achieve if you're eating a square of dark chocolate, which is 9 ounce, not 1 ounce is 9 grams of saturated fats. 1 tablespoon of coconut oil is 13 grams of saturated fat. So that already puts you up over the 22 grams. And then it's going to go into, then if you have some fish, which has both good and bad saturated and unsaturated fats in it, then you're going to start making cholesterol, LDL, from the fish, the fat in the fish. So you do need to look at that threshold, and sometimes people aren't quite paying attention to that. An easy way to negate that, just don't have any MCT oil, bulletproof oil, or coconut oil, and you've really dropped it down. Do watch, the dark chocolate. But a serving of steak for most individuals is anywhere from 3.5 to 4 ounces, or 3.5 to 4 grams of saturated fat, versus that 1 ounce of dark chocolate or 1 tablespoon of coconut oil, which is so much higher. So if we just sort of get back to real food, um, we can negate it. But from a idiopathic point of view, we have to look at the number of receptors to break down and then the production of the enzymes to recycle that LDL.
So, are there certain genetic patterns that you might see where to get the cholesterol panel looking within
Mhm.
range, shall we say, that you pretty much
Right.
have to go on a statin medication in some cases? Or is it
Yeah.
like maybe notwithstanding like, familial hypercholesterolemia or something like that, but are there certain like just very unlucky subsets where you're just probably going to need to go on a statin, or is that
I've had two patients in all of my years who they had to go on a statin
okay?
to bring it down, and then we just high dose ubiquinol with them.
Mhm.
To circle back to your HDL question, I'm actually okay. My HDL is 2.7, but my LDL is 2.
Mhm.
So, the bigger part is the ratio, as you know. And I actually had to fight with insurance companies for patients before because of HDL being really high, bumping it up over the numbers, even though their LDL was nice and low. And that is a more protective point of view. So I'm not too excited about that. It's generally a more protective point of view. Um, but there are some genes that are not in GeneRx called LIPC and things like that. And they talk about how much um, HDL you make for exercise. So if people who exercise a great deal, and I do, and I code normally for that gene, I make a ton of HDL through exercise.
Mhm.
I'm not going to say to myself or to someone, don't exercise to lower your HDL.
Mhm.
Um, the other thing with respect to LDL is fraction it out, right? Do the NMR test where you look at the particle size of LDL. Is it smaller? Is it large? What is the ratio? And therefore, what is the impact of this? And then I also say, get an angiogram and just see how much of it is it doing any damage? Because we know that if this is our blood vessel, what makes anything to glom onto it, cholesterol or calcium, etc., is stickiness. And what makes it sticky is inflammation. So we can really decrease a lot of the risk factors associated with higher cholesterol. Um, because I'm not a huge fan of bringing people's cholesterol down to zilch, which is what they often do in traditional medicine, and then cognitively they're struggling later on.
Mhm.
Yeah.
So just kind of a general question, in I'm paraphrasing here, but you mentioned or said something, I think a couple of times to the effect of, it's going to take, I think like 8 weeks or something to that effect to kind of like, reset that gene or kind of recalibrate that gene. Just so that we're all on the same page. I'm ascertaining that you're not insinuating that you're going to turn like, a T to a G or a C to an A. But would you mind just elaborating
Yeah.
on what you mean by resetting a gene?
For sure. So, every gene is like a light switch. It has an on and an off position, but that light switch always looks the same. It's just, is it on or is it off? So, if a gene is on, it's making that protein to do whatever. If the gene is off, it's not making that protein to do whatever. So, if it is a gene that makes a protein to make inflammation, I want to turn that gene off. If it is a gene that makes a protein to make more leptin for our metabolism, I want to turn that gene on. So it's really about triggering the expression of that gene to make that protein or to not make that protein.
And just to maybe get a little more hyper-technical here, because I just want to know for my sake, and for the most learned of listeners out there, like, are we talking about like gene like methylation or acetylation or things like that? Are we talking about transcription factor?
Transcription factors.
Okay,
Yeah,
okay,
100% transcription factors.
gotcha. Okay.
Yeah.
So I know our time is winding down, but yeah, I've got maybe a couple other questions I could ask, but are there other things about the GeneRx report that you think would be most useful to share?
Yeah. For me, if you treat all the areas that are in GeneRx, we're treating the underlying pathology of almost all disease. Whether it's you know, MS, whether it is the plaque in the brain, whether it's the low autophagy or the low mitogenesis that leads to those, whether it is cancerous in terms of PI3K pathways or mTOR pathways or autophagy or hormone stimulation. So even though the report doesn't jump out and say, you know, Lyme disease, this is what you do for it, or cancer, this is what you do for it. Um, all of the answers are in there. And often, as practitioners, when somebody first starts into genetics, it seems kind of overwhelming. You're like, oh my God, these alleles, what am I doing? And just go back to our foundation. We know what detoxification is. We know what it has, what impact it has on the body. We know how to treat it. We just got to see what's the strength within those systems. We understand the HPA axis. We understand autophagy. And so that's why, especially that's why I love lecturing with NDs versus any other group of docs because we have the foundation in this. Our whole education is about using natural supplements to, you know, manipulate genes. That's what we've been doing. And we just didn't know which gene we were manipulating and in what direction. And sometimes it worked and sometimes it didn't. Sometimes it backfired, those patients that had the adverse side effects. But the synthesis with the information is so much faster with our crowd and the educated public who is now also doing their research on this.
It's a very good segue into the next question I want to ask, or the after the next question I'm going to ask, because I'm sure a lot of folks listening are like, how do I access this test? So I'd like to get into that in just a minute. Just the one question that came to mind though, that if you don't mind my asking, so with the GeneRx test, it's your genes, so you really only, to my understanding anyways, only ever need to do this test once because it ain't going to change over time for better or for worse. I'm wondering, are there kind of peripheral tests, if you will, or adjunctive tests that you use regularly to kind of track progress, where if, the genes, and especially the way you have it laid out, which I, again, really, really appreciate, where it's very much like this, like action plan. It's not just, the blueprint and then like, what do we do with this information? It's very, very actionable, which is lovely. Um, with that being said, are you measuring people's, leptin levels? Are you measuring their, HOMA IR scores for insulin resistance? Are you measuring their, yeah, what are you measuring?
Absolutely.
Follow-ups.
So I do a bit of both. It depends on the patient. Certainly things like cholesterol or HOMA-IR or even just fasting insulin levels, like all of those, I absolutely. If it is cancer, I'm looking at the CT scans and things like that and requesting, those through their oncologist, obviously. If it is hypertension, we can look at their blood pressure. But for a lot of things, I don't use blood or saliva anymore. And the reason genetics is what's led me into that. So a really good example is I have very few B12 receptors. I am mostly vegetarian. I just don't like meat. It's just a taste thing. It's not anything else to do with that. I don't take a B12 supplement. I get little bits of B12 from egg. I do eggs and things like that. My B12 is always skyrocket high, but it's skyrocket high because I have very few receptors for it to be bound into. That doesn't mean I have good B12 activity. That means I have a lot of B12 useless hanging out in my blood, not doing anything. Because for it to have an effect, it's got to be bound into a receptor. So when we look at, neurotransmitter levels in the blood, when we look at a lot of different nutrient levels in the blood, it's the extra. So is it in your blood because you don't have enough receptors? Is it in your blood because you don't transport that nutrient or neurotransmitter to the receptor? Or is it low because it's all bound up into the receptor, having a really great effect? And that's where we have to marry the genetics. Okay, I see that you don't transport really well. And you do make a lot. That's why your levels are so high, but that's why you still have the symptomatology as if you didn't make any. Um, so it's really, genetics has really changed how I add extra testing into it. If it is a reliable test, and for it to be reliable, it also has to be longer term. Like TSH goes up 150%, 12 times over a 12-hour period. Not reliable. And, so we have to, again, I'm going to look at the gene that talks about how much TSH you're making on a daily basis. The normal amount, a lower amount, a higher amount, like that sort of thing. It gives me the patterns that I can then marry with the patient and their symptoms.
Makes a lot of sense. So how can folks access this test?
So GeneRx is a platform that is for healthcare practitioners only. So if you're not a healthcare practitioner, you need to go to your healthcare practitioner, such as yourself, and say, I'd like to do this test. And that's because there's a lot of medical information. We're talking about, altering people's serotonin and their dopamine, and I don't know if they're on a medication or not, and they may not know if it's going to interact with the medication that they're on. So I always want that to be overseen by a healthcare practitioner. The first step is to get your genes tested, get your raw data tested. The genes that I use are really unique. So there's only two tests. 23andMe Health and Ancestry, which has gone through its ups and downs. And it has to be the Health and Ancestry, or DNA Allure, which is a Canadian company based out in Toronto. So they have a test called the Ultimate Genomics, and that will both of those tests have all the genes that GeneRx uses. So you get those tested, send that text file to your practitioner, who then just creates an account with GeneRx and then drags and drops it into the account.
It is very easy on the
Yeah,
practitioner level,
yeah,
just to say. So yeah, once you have that TXT file,
yeah.
you're
Well,
good to
I'm a
go.
tech idiot, so it had to be easy. So I said to my tech guy, you need to make it so I can do it.
There we go. It's good. Well, I'm glad you set the bar low for that
Yeah,
because that's good.
I do. Yes.
Well, I really appreciate the time you spent chatting with me today, Dr. Kendall-Reed. It's been yeah, very, very informative. I'm really excited about the test. I've been starting to use it with patients, and yeah, just excited to use it even more after our chat today. Um,
Fantastic.
are there any social media platforms or online offerings or anything that folks want to work with you? Could you let us know where they can find you or anything you have for
So
offering?
I'm going to date myself here again. I don't have any social media handles. Um,
That's amazing.
and I sort of have enough ways of people being able to contact me. I am actually, as of July 1st, which will be 30 years in practice, I'm actually closing my practice to patients. I am only working with healthcare practitioners, helping them with their genetics cases. So, unfortunately, as a patient, they can absolutely get in touch with me on my website, pkrhealth.ca. They can email me and I can set them up with a naturopath that's near them to go through genetic testing and do this. But from a time point of view, because I already run the company and then I do a lot of lecturing and things like that, it was sort of my next level of getting the information out there at a higher level. Because I really do see a difference in treatment with patients when we do genetics and when we don't. So the more I can get that information out there by helping the docs, who can then help so many more people than I could touch personally, um, that's my next goal.
Makes sense.
Yes.
So GeneRx.ca, I believe, is the platform for docs to
For docs, yep,
access
genetics.ca.
the software, and then pkrhealth.ca for contacting you if they want to get in touch with the doc that works with you.
Exactly.
Awesome. Great. Well, I'll put that in the show notes. And yeah, thank you so much again for taking the time to chat with me today. It was very, very enlightening. I really appreciate it.
It was my absolute pleasure. Thank you for inviting me.
Well, we hope that you enjoyed this episode of the Overcoming Chronic Illness podcast, and please stay tuned for the next one.